Hello, everyone, and welcome to today's webinar, Elevate Your Research, Strategies for Effective Microplate Reader Utilization. I'm Cassie Saltman, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. First, I'd like to welcome our speakers, Dr. Leticia Montoya, Staff Scientist, Cell Biology with Thermo Fisher Scientific, and Andrew Dix, Staff Scientist, R&D, Protein and Cell Analysis with Thermo Fisher Scientific. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Leticia, Andrew, you may now begin your presentations.
Hi, my name is Leticia Montoya, and my colleague Andrew Dix is here today with me as well, and we will be talking to you about our microplate reader portfolio for Thermo Fisher Scientific and how effective microplate readers are to help you with your research. And so first off, why do you want to use a microplate reader, and why are microplate readers efficient? Truly, I think that microplate readers are a very powerful tool because they provide you a number of things. One, they're very high throughput, so it allows you to analyze multiple samples very fast at a robust rate. They're very versatile, so you can do absorbance, fluorescence, luminescence screening. There's alpha screening as well, which we'll get into later on in the talk. There's quantitative analysis, and so you're able to quantify exactly how much of your signal is being increased or changed over time due to certain treatments. They are automated compatible, so you can have a robot arm that allows you to take your microplates from one start of the lab bench and put it on the actual microplate, so you can microplate reader, and so you can do a number of microplates simultaneously over time. So it reduces hands-on time and it's very efficient. Additionally, microplate readers are very sensitive, so you can detect those subtle changes within your signal. And then data management is a lot less than if you were to do imaging. So imaging figures take up a lot of data storage, but microplate readers allow you to have that small storage of data, but also be able to see your data in real time as the microplate reader is reading it. And so with Thermo Fisher Scientific, we actually have a number of microplate readers within our portfolio. And these are just to highlight a few, from a microplate reader that only does absorbance to one that does absorbance not only in a microplate, but also in a cuvette. And then we have the VarioScan ALF, which can do absorbance, luminescence, and fluorescence. And then we have our VarioScan Lux, which is our most advanced microplate reader and has all the bells and whistles and everything that you need. And so depending on your need of what type of microplate reader you require for your assays, we have a variety that allow you to pick and choose. So as I said, microplate readers can actually measure a number of different types of light. They can do photometry, fluorometry, time resolve fluorometry, or TR-FRET. They can do luminescence, they can do alpha detection technology, and I'll get into a little bit of each one to kind of introduce you what are the differences between each of these technologies and measurements, and why would you choose one over the other. But before I do that, I do want to highlight what is the most commonly used. So the most commonly used method is photometry or absorbance assays. The reason is because there's a number of reagents that are available in absorbance. The second most commonly used assay is a fluorescence assay, and that's because it's more sensitive than an absorbance assay. But as you actually move down this triangle, you can actually see that the application just gets more specialized. And so they become less common depending on what your question is for your science. But as you go down this triangle, you get more specialized, and more particular and specific into the question you're asking to what reagent you're using for your research. So I'll work my way through down this triangle to walk through each of these assays. And the first one is absorbance, so photometry. So photometry actually measures how much light a liquid absorbs. And so with an absorbance assay, you definitely need a clear bottom plate. And the reason is because the light actually comes from the top of the well and goes through your sample, and it comes out from the bottom of the well. And so with that, the instrument itself is actually measuring how much light is being absorbed by that sample. And so the darker your sample is, for example, here on the right-hand side, you'll see that some wells are very clear solutions. So all the light, or most of the light, is going through the sample. As you get darker into that solution and the solution gets darker, less light is coming through that well and through that solution. So you're getting a higher response rate or a higher absorbance measurement as your solution gets darker. Next is fluorescence. And so fluorescence is a little bit different, where you have to excite a molecule in order to measure its emission spectra. And so with fluorescence, what you're doing is you're emitting the light that comes off of the compound. And so here's an example where we have a spectra, and this dotted line represents the excitation of that compound itself. So this whole entire spectra, you can actually excite anywhere on this whole entire dotted line spectra and get an emission from it. Now, you definitely want to excite at the most optimal point, which is the highest point of the peak. And so for this example, it's around 605, 610 is your maximum peak on this spectra. And from that, then you can get your emission spectra. And so that is going to be the solid covered spectra here, where you're measuring that emission of that compound itself. And so each fluorophore has its own characteristics in the excitation and the emission spectra, allowing you to multiplex multiple fluorophores at one time. Now, when we think about measuring fluorescence, there are multiple ways to measure fluorescence. One of them is through filter-based, and another one is through a monochromator on a microplate reader specifically. And so I do want to highlight this isn't only for fluorescence. You can use filters for luminescence and absorbance as well, but it's most commonly used when you're thinking about fluorescence. And so I wanted to highlight these and really talk about the differences between using a filter-based and a monochromator. So when you use a filter-based, when you use filters, it's very easy, it's very inexpensive. And the reason why it's easy is because each filter has its own bandwidths that you can actually measure within. So there's a bandwidth range that allows you to excite within that range, and then you can also measure the emission within a different range. But it does limit you. Because you have filters, you can't actually do a full scan of the whole spectra. So on the previous slide, I showed you the whole scan of the whole spectra. Well, with a filter, you can only measure the optimal excitation and emission of each of those compounds. Versus with a monochromator, you can actually do a full scanning. And so you can actually scan your whole entire spectrum of both your excitation and emission, because with a monochromator, you actually have the bandwidths of the whole entire spectra that you can see at wavelengths, throughout the whole wavelengths. And so you can do like each 2 nanometer read or 5 nanometer reads as you move along the whole spectrum. But it really just depends on the option that you need or the reagent that you need. But definitely before using reagent, make sure you know if your instrument is filter-based or it's a monochromator-based. So from that, the different fluorescence measurements that we can read with reagents that are available out there, you have a fluorescence intensity read, which is a normal fluorescence, which I described, where you have a fluorophore that's excited at 1 wavelength and emits at a different one. And then you have a dual label measurement, or what we know as FRET. And so with FRET, what it does is you have to have two molecules that are connected with one another, and you have a donor and an acceptor molecule. So when the donor molecule is excited, it actually transfers its energy to the acceptor molecule, and you're actually measuring the emission of that acceptor molecule. And so that's what FRET is that there's energy transferring from one compound to the next. Then you have time-resolved fluorescence. And time-resolved fluorescence becomes very specialized because you're using special fluorophores that allow you to excite multiple times. And because you're exciting it multiple times, you're also measuring the delay in the relaxation of that dye itself. And so you have to have this delay in time of measurement. And these are particular for specific reagents and assays that you are really interested in questions. All right, so from there, let's go on to luminescence. So luminescence is a little bit different than absorbance and fluorescence. Luminescence is actually a compound that emits light itself. So you don't have to excite anything. You don't have to measure any light that's being absorbed in the solution. It's just a compound that actually emits light off itself. There's two types of luminescence. There's chemiluminescence and there's bioluminescence. So chemiluminescence is purely chemical reaction. So it's a man-made chemical where you actually just emits light off by itself. Additionally, there's bioluminescence, which you can relate to, such as a firefly. So bioluminescence uses a biomolecule or an organism that can actually produce that light that comes off of the compound. All right, so that's absorbance, fluorescence, luminescence. Another technology that is commonly used is alpha screen and alpha ELISA kits. And so there's slightly differences between these two. First off, an alpha screen is very similar to FRET, but it is bead-based. And so with this, you have a bead donor and a bead acceptor, but on these beads, you have a molecule on each bead. And so on your bead donor, you have a molecule, say A, and on your bead acceptor, you have a molecule B. When those molecules come in close proximity with one another, the energy will be transferred from your bead donor to your bead acceptor, and then you're measuring the emission of that bead acceptor. And so this all has to do with the proximity of molecules specifically. So if you're looking at antibodies or analytes, do they interact with one another? You can see that interaction or how close they are by using an alpha screen. Now with an alpha ELISA, the advantage of this is that an alpha ELISA actually emits at a different wavelength than normally an alpha screen. Now, an alpha ELISA, the benefit of this is that it's very narrow and very bright. And because the emission spectra of an alpha ELISA, shown here on this graph on the right-hand side, because it's very narrow, you can actually run your assays in serum, plasma, cell lysates, anything that is complex and have a lot of proteins in it because of the alpha ELISA narrow emission band. So those are the differences. They work very similar in the sense that you have to have that connection between two molecules. All right, so we've talked about assays and we've talked about modes of read for light. Now, I kind of want to highlight the VeriScan ALF multimode microplate reader. The rest of the talk is going to be focusing on the ALF multimode microplate reader. It is our newest product that was most recently launched. It's very fast and flexible, easy to use. It does absorbance, fluorescence, and luminescence. The big advantage of this microplate reader, the ALF, is that you can incubate at multiple temperatures. So you can incubate from 2 degrees Celsius all the way to 45 degrees Celsius. You can shake on it. You can use a number of different plates. It's versatile in plates, so 6-well dishes all the way up to 384-well dishes. It does run with our ScanIt software, so any Thermo Fisher microplate reader uses all the same software, so there's no need to learn a new software. And we'll get into the software later on in the talk today as well. Before I get more into the ALF, I do want to talk about our microplate well washers, just real quick. So microplate well washers are amazing things that allow you to measure or to wash out your sample hands off. So we have a basic model that gives you 1 buffer bottle and 1 waste bottle, and then we have an advanced model that gives you 3 buffer bottles and 1 waste bottle. And so you can use multiple reagents with the advanced model. You can use multiple different types of washes, things like that. But this is all hands off, which is pretty nice and is very well equipped and able to use on 96-well plates and 384-well plates. Okay, so I've touched on a lot of these detection technologies and talked about each one of them. Now I'm going to focus on the ALF technologies, which is absorbance, fluorescence, and luminescence. With this, I do want to say that the ALF, or the VariScan ALF microplate reader, allows you to do solution-based assays. It allows you to do kinetic reads, endpoint assays. You can even do, you can look at individual cell-based assays. And additionally, you can look at nucleic acid or protein quantification and the spectrum of those. My colleague Andrew will get into specifics of that and how you do that on the ALF. Now, with the assays themselves, I want to touch on a few of them. One of them is the absorbance assay. This is the most commonly used absorbance assay, Cyquant XTT. And Cyquant XTT, it's an add and read reagent. So you add it to your cells, you incubate at 37 degrees, either in the cell culture incubator or on the microplate reader itself. And then you can read the absorbance from that. The way that this assay works is that you have an electron mediator and you have a redox dye. The electron mediator is PMS, and the PMS actually scavenges all the electrons to the plasma membrane where it interacts with the XTT to reduce it to give you this dark orange pigment product. And so the darker it is, the more viable your cells are, the more healthy you have, the more healthy cells you have in your well. It can actually detect all the way down to 100 cells per well, so it's somewhat sensitive, but we do have other assays that get more sensitive, like fluorescence assays. Before I move on to our fluorescence assays, I do want to show that you can actually quantify each of this. So this is an example where we took cells, we treated them with gambogic acid with a number of different time points, for treatment of that drug for different amounts of time. You can see that the IC50 changes over time as you increase that drug incubation time. So if you're looking to get more specific or more sensitive, I would use a fluorescence assay. And this is an assay that actually does absorbance and fluorescence. So this is Presto Blue HS. It's a derivative of our Presto Blue product. The HS stands for high sensitivity. What we did here was we were able to be able to purify the compound even more so that you can actually get a higher sensitivity of your cells and your viability of your cells and the number of cells that you're actually detecting in a well than you would with our original reagent, Presto Blue. The way that this assay works, it's an added read, so you can add it to your cells, incubate for some time, again, either on the microplate reader or in the cell culture assay or in the cell culture incubator, and then you take a reading both either absorbance or fluorescence. If you want higher sensitivity, use fluorescence. The way that this assay works is that you have rizazirin, and so rizazirin is blue and lowly fluorescent. So in this picture, you can see that all the blue wells, you don't have very many viable cells or metabolically active cells. The rizazirin will then react with NADH, which then gets reduced to resirufin. And so the resirufin is actually what you're measuring when you're looking at fluorescence, because that resirufin is not only pink, so you can visibly see it, but it is also highly fluorescent. And so we can actually detect down to 20 cells per well when you're using the fluorescence mode of this assay. And so you can see on the plate depicted here on the bottom left that we have a range of cells or wells that are pink and kind of purplish. And then we have far right, there's blue wells that are non-fluorescent, where we don't have any metabolically active cells. With this reagent, we can quantify. You can quantify, this is an example where we took cells, we treated them with a drug for different amounts of time, and we were able to see that IC50 change over time. Additionally, we were able to see that if you're measuring fluorescence over time, you can see that your fluorescence maximum is around 4 to 5 hours. After that, you start decaying in fluorescence, which is common for this type of product. If you let it incubate too long, the resirufin will actually over-reduce and you'll have a non-fluorescent compound. So with this assay, definitely make sure that you don't incubate for too long so you don't have a false negative. All right, so next up is luminescence. Here is a luminescence assay. It's very high throughput. It does do cell viability screening. And the way that this works is it actually quantifies the ATP that's present within your active cells. And so when ATP is present, it interacts with the D-luciferin, and you get light that comes off of your compound or your solution itself. You can measure this. It's very sensitive. I will say that luminescence is the most sensitive assay that's out there. So if you're looking for a very highly sensitive read, then I would use a luminescence assay. Okay, so with reagents, you also got to be able to work up your data. And so when working up your data, you have to use there's a number of, you can use Excel, you can use a number of different, softwares out there. But ideally, you would want to use the software that your microplate reader uses and that it comes with. And so the VariScan ALF actually comes with a ScanIt software. And this ScanIt software allows you to work up your data at the very end. But in order to do that, you kind of got to go through a few steps. So the first step is actually your plate layout. So you got to assign and label your wells with your experimental design. Normally we run things in triplicate, if not more. But this is an example where we have triplicate of a drug dose response. After that, you run your assay. So you get all of your data filled into your wells. From there, you can analyze your data by obtaining the average of your replicates. So this is the average of all 3 replicates on the top row. Then you can subtract out the background, or your AKA blank. So you definitely always want to make sure there is a blank, especially depending on what reagent you're using. Subtract out that background in order to get a true understanding and readout of your experiment. From there, you can normalize your readings. Normally, I normalize drug dose responses so that you can normalize it to your highest response that has come out. And so you'll notice on column 11, that is 100% because that is my highest readout from this assay itself. From there, you can do a drug dose response curve. And so if you click on the results, you can click on drug dose response. You can give your chart a title. The next part, you definitely want to make sure that you switch your x-axis to logarithmic scale so that you can go from a non-fit curve to a very fit curve in log scale for your drug. From there, you determine what graph line fit you want. So there are a number of options on the ScanIt software. Pick which one is best to your your reagent or your question that you're asking, or what drug you're using. From there, you can establish your IC50. And then from there, you can observe your percent CVs and your fitted signal. And then you can label your axes so that you have a graph that you can present and publish in a paper. All right, so I talked a lot about reagents, the differences between reagents, what we can do on the ScanIt software. I will say that there are a variety of microplate reader applications. So the world is your oyster. Pick a number of reagents. Our VeriScan ALF can detect it and read it. We have ELISAs, we have DNA and RNA quantification, which Andrew will get into, enzyme assays, apoptosis assays. There's a number of reagents out there that we provide that do match with our ALF. Additionally, to highlight the ScanIt software, it can do automatic dynamic range selection, so you're not having to guess what gain you need in order to detect the most optimal signal. It does an endpoint assay. You can do kinetics, you can do absorbance spectra, even fluorescent spectra. You can do parallel line analysis, and what that means is that you can actually compare two plates with one another, so you can look at two data points at one time on one graph. So ScanIt software is very versatile and very user-friendly. Additionally, what's new on the ScanIt software, we do updates on the ScanIt software on a regular basis. So what's new on the ScanIt software is that you can actually plan your experiment in an Excel sheet and then import that and auto-load it into the ScanIt software so you can run the same assay over and over and get results very fast. Additionally, we do have a fluorescent spectra viewer tool. So if you pick a fluorophore, it will actually give you an excitation and emission spectras for those, so you know what the excitation and emission wavelength optimum is. It is a no licensing fee, so you don't have to pay for a license for this software ever. It's one-click data export to Excel if you really want to go in Excel and look at things later. It's user-friendly, simplified for a user firsthand. One more product I want to talk about is our microplate, our micro drop plates. So we have micro drop plates that allow you to measure samples that are anywhere between 2 and 10 microliters. So maybe you don't have enough sample to fill a 96-well plate or 96-well dish. You can use these micro drop plates to measure on the lower end of your volume. And with the micro drop plate itself, you can do 16 samples. If you get the microdrop duo plate, it's up to 32 samples. And so it fits like a 96-well plate in the actual instrument. You can also use this on the VeriScan ALF. The best thing to use this for is actually determining nucleic acid concentration or your protein concentration, and look at different absorbances for those. Now, with that, I do want to introduce my colleague, Andrew Dix, where he will go into our quantit assays, looking at nucleic acid quantification, and he'll get into depth of what these assays do, how you can use them, and how they pair up with our VeriScan ALF microplate reader.
Thank you, Leticia. As she said, I'm going to be doing a deep dive into the application as far as nucleic acid quantification, primarily focusing on our quantit assays. So nucleic acid sample types can come from anywhere and everywhere, whether those be cells, plants, tissues, bacteria, cell-free forensic. And what you do with those samples is you may be going into PCR, you may be going into sequencing, but you want to focus on that experiment. You want to get into that experiment with confidence so that you know that you're investing your time appropriately. And that's where we come in. And we want to be able to offer that through our quantification efforts, which is not necessarily where you want to spend your mental energy focusing on. So we're trying to streamline these as best as possible for you. And we do that with our quantit assays that are specifically formulated for microplate readers, or rather fluorescent enabled microplate readers. So here, the ALF detector or the ALF system, as Leticia was overviewing, or the Lux, which is going to be bigger brother, so to speak, that has a little more options towards it. And so the quant assays, there's a variety of them. I think there's about 18 in total. They're going to be focused on nucleic acid quantification in general. We also do have protein and endotoxin assays, though. But that core set of assays is going to be on DNA and RNA quantification. And the different flavors of those assays are going to be focused on whether that be detection range, so HS for high sensitivity, which would be a lower detection range. So, for example, the DNA HS assay is going to focus on 0.2 nanograms to 100 nanograms of the detection range, whereas the BR, or the broad range, is going to focus between 5 nanograms of input to about 1,000 nanograms of input. The same thing will correlate for the RNA assays, going from a high sensitivity range to a broad range, and then to finally an extended range. And looking at the graph here on the left, we can see where the UV-Vis range would be captured, and fluorescence in general is going to be able to go to a lower detection range, again, as Leticia was overviewing for the cell health assays. And as all of these are fluorescent assays, they're all going to be a no-wash fluorogenic style approach that can be used in a 96-well or a 384-well microplate format. And all of these assays are going to offer the accuracy and the reproducibility that we are proud of with our quantum assays. So that's going to be greater than 85% accuracy, or to think of it a different way, less than 15% error from your expected value. And the reproducibility, as measured by the coefficient of variation, will be 5%, can be as low as 5% or less. While we do offer a variety of kits, they do come in 3 core different styles. So the main style of our quantum assays are going to be what I refer to as the traditional or classic format, and that's going to be where your buffer and your dyes and your standards are all being offered as separate entities, and they're all going to require the user to do something with them. And so for those assays, you're going to have to first prepare your working solution, which is what we call the mixture of your dye and the buffer. So once that's prepared, you're going to, on the microplate, combine the standards and samples with that working solution, then incubate that for several minutes, and then read that on an appropriate fluorescent detecting device, whether that be the ALF detector, a lux detector, a qubit, or what have you. One of the innovations we've made over the last several years is offering the 1X system. So the 1X system is currently only offered for the DNA high sensitivity, or HS, and the DNA BR system. In this case, we've done step 1 for you, where you no longer have to prepare the working solution. We've done that for you in a very stable fashion, where all you have to do is mix that on the plate reader, mix that on your microplate with your samples and standards, and incubate and then read. That's going to be incredibly useful for folks doing a lot of high throughput. So if you're measuring a lot of plates and you want to decrease your workflow and increase your confidence, this is something that I would strongly recommend. It's also really nice for automation style workflows, because you can take that working solution, have the confidence that it's stable, and have the confidence that what you run for your first plate at the beginning of the day is going to be consistent with what you're running at the end of the day on your last plate. The last format that we offer is going to be the ready plate format. This is a relaunch of something that's been existing, but we refreshed it to give it a little better feel given the expectations of the modern user. So here we actually combine the buffer and the dyes on the plate itself in a dry down fashion. So the user simply needs to add either water or their sample, incubate, and read. And this is the most streamlined workflow that we offer, although at this point it is only available for the Pico Green and the Ribo Green systems. Now, all of those formats and all of those different styles can be read on the filter-based Veroscan ALF reader. They can also be read on the Lux, which is again going to be the monochromator version, which has a few more options, or bells and whistles, as they say. And here we detail out the different filter pairs that we recommend or that would be required for the different assays, as well as the monochromator settings. And regardless of which setup you're using, you should be able to get that all those things that I mentioned before with respect to accuracy, reproducibility, and detection limit. It's not going to matter whether you're using a filter base or a monochromator system for these assays. Some of these slides may look a little familiar, so this is just continuing off of what Leticia was saying about setting up your assays in advance using the ScanIt software. So ScanIt is going to run all of the Thermo Fisher style microplate readers, which is really nice to have that consistency. It's actually an incredibly powerful software that can do a lot for you, provided you give it the information that it needs to run. So what we can see here on the left side is the first screen you'll have. So when you set up your plate, if you include in the green style color, if you label out your standards, so all the quant assays are going to come with 8 standards. So if you include the 7 that actually had sample in them, if you're using the DNA HS again as an example, these are going to be the 0.5 nanogram per microliter samples to the 10 nanogram per microliter samples. So if you detail those out in triplicate on the left side, and if you save that bottom and specifically call that your blank, so this would be your standard 8, standard 1. I actually always mix up that order if 1 is the blank or 8 is the blank. So if you include the blank as a separate denotation, the system knows how to handle that and can then do a really neat function later on. So if you have those 8 on the left side and then you reserve the right side of the plate for all of your unknowns, you set up your plate. And then from there, if you go to the add step on the next screen, so this is going to be the screen on the right side. If you do the add step, add fluorescence, select the filter pair that would be required for your assay. If it's a DNA assay, this is typically going to be the blue-green filters that are highlighted here on the slide. If it's an RNA assay, it's typically going to be a red and far red filter pair. From that, you're actually ready to go. It's really easy to set up these assays on the ALF system because it is so streamlined. So from here, you would hit that play button in the bottom left, acquire all of your data, and then we'll get into some processing tips. So processing, Leticia mentioned this a bit, but I want to go a bit harder into this. So one of the first things we recommend doing is doing that background subtraction. And since you told the system where the blank was, it can do that background subtraction universally across all the data that was acquired. So that would be the first thing to do. The second thing to do would be to do a curve fit. And if you hit no other buttons other than those 2, this is the data that you'll get out. So in the core of the screen, you can see on the green, I actually had 6 replicates here just to show off a little bit. And then I took that lowest standard, that 5 nanogram or 0.5 nanograms per microliter sample, diluted it further to get 2, 1, 0.5, and 0.2 to express that last chunk of the detection range for this DNA HS assay. I wanted to show how that worked when we look at the percent error and the percent variance, and all that's going to be captured on the right side of the screen. The lower half that wasn't the traditional standards is going to be on the lower half of that table. So if we look here, having clicked nothing else in the software, all of the data and the percent errors look amazing for that core where the samples are, where the standards are represented. So from 100 to 5. That looks amazing. However, the system doesn't know how to handle that data below the lowest standard and between that and the 0. So you get a lot of NA or less than min, which is quite frustrating because we tell you that it can calculate that, but the system's telling you no. And that's because you have to do a couple extra things. So with that, what you have to do from here is you need to click to force through the origin. And you can do this because you did a background subtraction. So you can do this with confidence. And this effectively says 0 is 0 because I told you what the blank was. We've corrected for that. And so we can do this with confidence. And then we'll click extrapolate, which is going to be sort of the middle of the left panel of your screen. And this will allow you to access that lower portion of the data, which is below your lower standard. So below that 5 nanogram sample. So now we can see that we get really nice data for that entire detection range. I also wanted to point out that with respect to the standard curve on the far right side of the screen, I personally always like to see the replicates and where the unknowns are. I find that that visual aid really helps center how confident I am with the experiment. If all of my unknowns are at the lowest low end, then that informs me that maybe I should add more sample, or maybe I should have another standard. If I see that those red bubbles where my standards are have a lot of variance, and don't stack really nicely where you kind of actually can't tell that there are replicates, that suggests that I have some outliers and I might need to do a treatment there. So please pay attention to that. It's some extra little bits that the ScanIt software can do for you to help make your data a little more visual and a little cleaner workflow. Now, biology doesn't tend to be linear, and neither are a lot of the assays. While they will be very linear over the range encompassed by the standards, what happens below that or above that will be nonlinear. And in order to be more robust, we strongly recommend using a nonlinear algorithm. In this case, the ScanIt software has a 4PL built into it, which is an incredibly powerful algorithm or curve fit. It will do great things for your quantitative assays. So if you have to use linear, please make sure you force it through the origin and do a background subtraction. If you are able to use nonlinear, I strongly recommend doing those previous things, the background subtraction, forcing through the origin, but also doing the 4PL. And if you do that, make sure that you do click, again, the extrapolation, and you go into that advanced setting below. So the middle of the page, the lower bar highlighted in orange, you're gonna have to change linear to logarithmic. And that will give you some incredibly tight data with respect to your dynamic range and give you a little extra flexibility at that higher end and lower end for those assays that aren't explicitly linear. And so from that, I want to transition a little bit. So we've talked a little bit about the different types of quantitative assays. We've talked a little bit about how to run your assays on the ALF system, or actually any ScanIt system, really. But I want to talk a bit about how to get the most out of your assay when you're actually setting it up. And a lot of this detail is covered in various app notes that are available on the web, which I'll have links to later on in the slide deck. But I want to go through some highlights. So one of the things that I always like to let users know is pipetting and mixing order. They're pretty related. Most users we find are talking between 1 and 2 microliters of sample, and oftentimes don't necessarily appreciate that there's a lot of error that goes into that initial dispensation. So if you're using a P10 system, if you look at the left graph, highlighted in orange is going to be if you're using actually a P2 pipetter. So all those different dots capture a different measurement. And so the tighter the dots, the lower the error and the better the sample-to-sample variation, or the tighter the sample-to-sample variation. So tighter is better. So we can see that the best performance here is going to be using a P2 pipetter pipetting 2 microliters, followed by 1 microliter being dispensed with a P2 pipetter. And if you're going to be using those low volumes, please don't use a P10. You're going to get a lot of error that you're not necessarily going to know what to do with, and it's going to show up in your results later on. So while you can physically do it, we don't recommend it if you need precision. And again, there's more information about that online. The next thing I want to talk about is mixing order. So the protocols are written where technically they say add your sample onto the working solution. So you would first plate 200 microliters of working solution and then 2 microliters of your sample on top of that. If you're doing really strong pipette mixing, that's incredibly acceptable. You're going to get fine data. But I find when I'm mixing with a 2 microliter pipette, trying to wash out that last drop, I don't have enough oomph, if you will, to mix that entire 200 microliters. So in order to get that done, you need to be really aggressive with a plate mixer or come in with another pipette that's set at a much larger volume in order to get a really vigorous, thorough mix to have that nice sample variation you'd expect out of your results. So in order to do that in a more expedited way, I recommend adding the sample first. You don't necessarily get to rinse out your sample, but you do get a lot of you do get a lot of that, you do minimize a lot of that variation, which is incredibly nice. So by adding the sample first and then your working solution, you allow the solution dynamics to do a lot of your mixing for you, to the point where if you don't mix it all after that, you actually still have comparable data to if you had added your working solution first, then mixed. And so one of the next things I'd like to highlight is, I mentioned this a little earlier on, is lowering the lower end in the standard curve. So if you find visually, if you're looking at that scan it style graph with those gray points where all of your samples are, if you find that you're routinely at the lower end below the lowest standard, again, in this case, for the DNA HS assay, that'd be 5 nanograms or below. If you find that you're in that range, I really recommend populating that space with an extra standard or two. And shown on the right in red data points, we show what that might look like if you take that 7th standard and dilute it using a little extra buffer. That it's not required by any stretch, but it might give you more confidence when you're working with samples in that range, so that you can see how that's affecting your standard curve, or how your standard curve choice might affect your data integrity at that point. With that, we did talk about how linear is not necessarily always the best. The ScanIt software actually prohibits you from extrapolating in your linear unless you ask it to. But if you are using a program like Excel or Prism or Jump, you can do that and you don't necessarily have the same warning signs. So what we wanted to highlight here is just to drive home that if you do linear and you don't do anything else, you can have as much as 50% error on the low end. And that's because the way the curve fitting works is it takes that top end and it fits that first and prioritizes that first, and then slowly goes to the lower end. So your last sample has the least priority, and so the least impact on your R squared, or your correlation coefficient. So you might have an amazing R squared, but then some really terrible fit at the bottom end, or it might even be negative, which is just odd to have negative fluorescent signal. And so what we strongly recommend is to do that background subtraction, force it through the origin, and to do something nonlinear. And when you do that, you'll have data that really nicely, we feel, complements your data, the integrity that we expect and that we hope that you're able to obtain. And if you're coming from a qubit system, it's going to best mimic that type of data. Lastly, I wanted to point out how to get the most from your samples with respect to throughput. So all of the quant assays are amenable towards liquid handling techniques. We just want to point out that, at least from my experience, I've learned that it's not as simple as walking up and wanting to use it that day. There's a lot that goes into it. We could give whole entire seminars on how to use them best, but they are all amenable to those workflows. I do personally think that the 1x solutions are easier to use with some of the higher liquid handler throughputs. But if you do want to use them, I do recommend, again, adding that small sample first. So adding your sample first, then adding the working solution so that you get some of that mixing dynamics done for you. And you don't have to take all of these plates that you just set up, mix them incredibly thoroughly, and then get to your reader. It just creates a bit more of a backlog, which is the opposite of what you're trying to get at when you're working with a liquid handler. The last thing with respect to throughput is all of our assays are amenable towards a 3 to 4 well workflow. So if you need to be using even lower sample volumes than 2 microliters, you can augment your 96 well workflow with a 3 to 4, where you can use those same standards that we're offering or that come with the quant assays, add 10 microliters of that. But then what you'll do with the working solution is you'll actually add a more concentrated amount of dye. So rather than a 1 to 200 dilution ratio that's prescribed in the 96 well plates, we're going to actually increase that by 2 to 5x, so that it's going to be a 1 to 100 or a 1 to 40 level dilution. And when you add this at 30 microliters, for example, that's what I use, you can get really nice data that's captured here on the right, which will offer the same detection range, the same confidence, and the same R squared that you'd be able to see with your 96 well format. And so there are additional resources for all of these on the web. The cell health assays, we've highlighted a few app notes here, as well as a few links to help you get more out of your protocols and out of what you're working with. The quant assays, again, highlighted here. There's going to be more detail on everything that I just mentioned, as well as some links to check if you need additional resources that you can't find there. We are here to help. We want you to succeed, and we want you to get the most out of all of these experiments. So again, if you need anything, please reach out, and hopefully we can help you find the resources you're looking for. And so lastly, we know your time is precious. Leticia and I just want to say thank you. So thank you from us, from Thermo Fisher. We hope this helped. If you have any questions, again, please reach out in the chat right now, email us later, or reach out to your technical assist representative. And with that, Just want to say, lastly, thank you again. Happy sciencing and good luck.
Thank you, Andrew and Leticia, for your informative presentations. We will now begin the live Q&A portion of the webinar. We've had a lot of questions that have come in so far, but if you haven't already submitted any questions, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Our first question here, and I'll just remind Andrew and Leticia if you want to come off mute, perfect. Our first question is a quick one. It says, I may have missed it, but you used the phrase force a good origin. Could you expand on this, please, or just give a quick repeat?
Can you hear me okay?
Yes, we can hear you.
Awesome. I'm assuming this is for me and not Leticia. So if you do the background subtraction, I will force the curve fit through the origin. And that makes sure that you're taking advantage of y equals 0 because you subtracted off your background. So no signal should be 0. So you're just forcing the math to do that accordingly. Otherwise, you get some oddities in that curve fit where you can have your background actually show up as negative, which is confusing and not good. But yes, so hopefully that's clear. If not, just please leave another note in the chat and I'll try and maybe email you and maybe that'll be better. Leticia, anything to add if I just sounded confusing?
No, I think you're good.
Awesome.
Yeah, reach out if that wasn't clear, but happy to explain more.
All right, moving on. Our next question here asks, what are some common challenges or pitfalls in using plate readers for cell health assays and how can they be mitigated?
So I don't know, Andrew, do you want to go first? Do you have something in mind, or
this is
you want me to
self-help
take a stab
essays,
at
right?
it?
This is you.
All right, well, I'll take a stab. So I think some common challenges and pitfalls are really just understanding that you have to have a blink or a background well for your cell health assays. Your reagent itself normally is, has some either type of fluorescence or luminescence background or absorbance background that you need to subtract out of your samples. I would say that would be the first thing. So you always want to have a few blank wells for your reagent, specifically in the exact same solution as what your cells are in, whether that's PBS or media. So that would be my most one that's I would call out. I think a second one is optimizing your reagent for your cells that you're using. So with cell health assays, you always kind of want to stay within the linear range of the reagent. If you go beyond the linear range or you're in the background of the reagent itself, you're actually then either get false positives or false negatives. So make sure that you do some type of standard curve before you continue screening within that reagent. Those are probably the top two.
Wonderful. All right, let me go ahead and move that over. Our next question asks, what type of plates should I be using for cell-based assays?
Yeah, I think that's a good question. I think it depends on the cell-based assay. So if you're using a fluorescence assay, you definitely want to use a black well dish. If it's absorbance, you definitely want a clear bottom, but for fluorescence itself, you want black wells. For luminescence, I would say using a white well dish, whether that's a 96 well plate or 6 well dishes or anything like that. And then for absorbance, you can either use black or clear, but either one will work. So it depends on your assay and what cell-based assay you're using. It all has to do with how the light actually gets absorbed or excited off the plate or repelled off of the plate. So Black for fluorescence, white for luminescence, and clear black for absorbance is the general rule of thumb.
Okay, great. I think this next question here is maybe for both of you. This asks, when should I consider a micro drop plate for nucleic acid quantification versus quant reagents?
Leticia, do you want to go first or shall I?
I've answered two. You go for it, Andrew.
Rock, paper, scissors rules. That's how it is.
Great.
So neither's wrong, and it's more of a personal preference or what you know about your sample. What's really nice about the micro drop plates is that there's no consumable, so it's very much like the nano drop where you're not generating any plastic tubes. You're using your sample directly on a plate that you can rinse and wash off and reuse. It's really nice when you have a lot of sample, a known high quality sample. And when I say a lot, I mean volume and concentration, because UV is going to have a higher detection range, which I think I showed on one of the earlier charts. Fluorescence is going to have a broader and lower detection range, and it's going to be able to discern whether you have any impurities. So if you don't know the quality of your sample and you don't know the concentration of it, that's when I would recommend a quant assay and using fluorescence. If you know you have high quality sample and you know you have a high quantity sample, then using something like a micro drop plate would be a perfectly reasonable and straightforward thing to do. So I hope that helps.
I agree with all those. Yeah.
I love it when we agree.
Awesome. All right. Leticia, anything you wanted to add before we move on to the next question?
No, I think Andrew hit on all of them. I think it has to do with more of your output and, once you get the micro drop plate working, I think it's amazing and great because you can do a number of samples at one time in a fast, with low volumes. Yeah.
Awesome. Our next question here, Andrew, is for you again. Can quant assays have the same performance as a qubit?
The short answer is yes. The longer answer is it sort of depends on the user. So they're designed to be comparable and they're designed to offer the same amount of performance. One of the things we strive to do with the qubit system and the qubit workflow is to make it as user-friendly as possible and to take a lot of that workflow calculation and provide it for you. And in our opinion, that's what customers who are going for the qubit want to see. With a quant, because as Leticia mentioned, we never know necessarily what plate reader system you'll be using. You have the freedom to use any. It's democratized in that respect. So that isn't done for you. And that's where a lot of that error or discrepancy would come in. It's not that one's better or worse. It's just that there's more freedom in the quant assay. So if you do choose to do something like a straightforward linear fit with no additional caveats, you will not get the same quality as the over-engineered curve fit system that we have in the qubit. The qubit has special algorithms for all the different assays that are based on an innumerable number of replicates, different users, different times, different lots, things of that nature. So it's going to be more robust. When you're doing a quant assay, again, you have that freedom, which is why I would really stress to do background subtracted, force through the linear and force through the origin and do something nonlinear. It'll give you that additional freedom and it'll best mimic the qubit, but it is an ongoing struggle we feel for folks that want to do higher throughput, which is more quant oriented. But just keeping in mind that with that freedom and with that ability to optimize all that on your own, that sometimes it does require a bit of optimization on your own. And so there are a few things to think about as you go through it. So again, hopefully that was clear.
Yeah, thank you. All right, we have time to squeeze in maybe just one or two more questions here. We're down to the wire. So what types of plates should I be using for quant assays? Our next one.
So Leticia said this pretty well when she was overviewing sort of a rule of thumbs with respect to different types of plates. I would prefer using black walled plates, flat bottom, and if they're clear bottom or also black bottom, that's sort of a dealer's choice, as it were. I tend to use top read fluorescence, so either type of plate would work. If I can only buy one thing and I want it to be the most versatile, I will use the black wall clear bottom flat plates, which I think we can put a link to in the chat, because you can use them for absorbance or fluorescence. You can use them for top read or bottom read. So that way, if I'm stocking up a whole bunch of one thing, in my opinion, that's the best one size fits all. Luminescence is a different beast, which Leticia again mentioned. And that's just a pretty different workflow.
Okay, great. And Leticia, we'll throw the last one over to you here before we go. This question asks, does Veriscan ALF have 21 CFR Part 11 solution?
It does, actually. So the 21 CFR Part 11 actually comes with our software. So the ScanIt software that we talked about today in our talk, there is a version of it that's 21 CFR Part 11. So it's not necessarily to the plate reader itself, but our Veriscan ALF is compatible with our software that does have that Part 11. So, yes.
Wonderful. All right. Thank you again, Dr. Leticia Montoya and Andrew Dix, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.